Biosimilars are biologic agents that are biologically similar to a reference product that's already been approved for a specific purpose. These are large molecules that are made in living cells, and there's a complex process to their production that includes a bunch of things being added onto it, post-production modifications such as adding sugars and things onto the agent. And for this reason, there has been a lot of discussion about how you would ever make an agent that wasn't the original reference product or the original branded product for that biologic agent. And in the spirit of competition and trying to make additional agents available after the patent life wears off runs out, specifically on a particular biologic agent, the concept of biosimilars arose. And these are biologic agents made in living cells that are similar but not identical to the original reference product. And these agents are defined as having no clinically significant or meaningful differences in efficacy or safety, along with multiple other factors that have to be similar and safe.
A biosimilar has to have the same or similar route of administration, of storage conditions, as well as of the strength of the drug as has the reference product. The manufacturers have to show this during the development program of the drug before it can be approved, that these conditions are similar. The difference between generics and biosimilars is easily understood if you imagine how they are being produced. While generic medicines are usually chemically produced and can easily be reproduced because of the chemical formula you have, there is no big effort actually to get these approved, and you don't need large clinical trials. However, biosimilars are being produced by living cells. So every batch differs to a certain extent from the other, which is if there is no change in activity of the substance, no issue, and is being tolerated by the agencies responsible for the approval of the drug. It is necessary to actually not only do a lot of preclinical analysis, but also clinical trials to see that there is, based on the totality of evidence of these experiments and preclinical and clinical studies, that there is a highly similar product available. This can lead then to the approval of the drug. However, it is much more complex than just produce a generic compound.
Generics are usually small molecules, and it's, make the example of aspirin, that basically, or ibuprofen, where you have a small molecule. It's a chemical structure. You make an identical chemical structure, and you show that basically there's no major differences in pharmacokinetics or purity in an initial small trial in healthy people. And once you've done that, the agent basically can be marketed as a generic. And actually, the chemical structure just needs to be shown to be the same. And in some of these, you just really need to show in animal models that there's no differences in terms of the pharmacokinetics or impurities. So you have to show it doesn't have impurities in it. But that leads to a whole lot of different generics out on the market, made in different countries with different binders, etc. But they are the same chemical structure, so they're thought to be the same in terms of their clinical effect. If you're looking for ibuprofen, whether you get one that is a generic or a branded name, it shouldn't have a difference in activity based on this long-standing definition and approval process. But biologics are different. These are very large molecules and usually monoclonal antibodies. They're made as we've discussed in living cells, and there are different processes that are involved in creating that biologic agent that are quite different from making a simple chemical structure. So you have to have the antibody, but then there are these modifications to the antibody, something called glycosylation or adding sugars on. You have to make sure that the drug doesn't cause antibodies. So, it's an antibody itself, so some of these actually can be immunogenic. So there's a lot of different ways of looking at biologic agents that are completely different than the way we look at generics or small chemicals and small chemical products. Then, of course, you want to make sure that not only is it similar in terms of its structure and its in vitro measures of efficacy, none of which are perfect for biologics, but you also want to make sure clinically that it's similar as well. And that's where you get the whole concept of biosimilars. Very different, very complicated process of demonstrating that these are similar to the reference product.